also, and some substances which are hydrophobic. They can easily pass the lipid bilayer. But those who are hydrophilic, they cannot cross the lipid bilayer. Examples of such substances: hydrophilic substances, water-loving, water-soluble. Cell needs so many things for its survival. So from where it will get? From outside. What are the things they need? Say glucose. It is water-soluble. These are required by the cell because cell has to perform so many functions, and cell is having, and for functioning they need enzymes. Now these enzymes should be present inside the cell, and where from this enzyme will come? Cell, where from it will get? Because in the nucleus there is DNA, there is genetic code for these enzymes, and this genetic code will produce mRNA. This DNA will produce mRNA. This mRNA will come into the endoplasmic reticulum. There, the protein enzymes are protein in nature, so this will be synthesized in the endoplasmic reticulum. So in the endoplasmic reticulum it is formed. Then they will be released. They will be going to Golgi bodies for packaging and processing. Thereafter, it will form. These enzymes will be encircled by a membrane as zymogen, inactive enzymes. So whenever there is needed, enzymes are needed, so they will be liberated from this package and will show its activity. Okay, so to perform its activity, enzymes, they need the substrate. Suppose cell has to synthesize hormone. What type of hormone? Say steroid hormone. So steroid hormones are synthesized from what? What is the raw material? Raw materials are acetyl coenzyme. Acetyl coenzyme. From this material, actually, the skeleton, basic skeleton, cholesterol is having a basis. Cyclopentanoparahydrophenanthrin ring. These rings are formed. Then, with addition, deletion of certain groups, some modification in the skeleton, they produce different steroid hormones like estrogen, like progesterone, like corticosteroid hormones, like glucocorticoid hormones. So in this way, they are produced where? Within the endoplasmic, smooth endoplasmic reticulum. So raw material has to be supplied from outside to inside. Okay? So transportation is needed. Again, the cell is metabolizing something, or it is synthesizing, say, insulin in the beta cells. So insulin, beta cells don't require insulin. This is required by the mesosomatic cell, other cells. So they have to come out of the cells. So they will be coming out of the cell by the process of exocytosis. This is insulin, glucagon. Okay, so who will help in transferring this material, synthesized material, for going outside? The cell membrane. The cell membrane contains various receptors. We have seen yesterday. Various types of receptors. Some receptors, there are receptors, there are channels, there are carrier. Okay, for example, receptors. Receptors remain outside the membrane, attached to the membrane, and the receptors also having some other part, another part, enzyme part. So they will remain inside the cell towards the cytoplasm. So now the hormone will come through blood, will enter into the extracellular fluid. From extracellular fluid, it will come closer to the cell membrane, and there it will find receptor for it, specific receptor. So it will look for, say if it is insulin, so it will look for whether insulin receptor is there. So it will bind with that receptor. When this receptor became bound to this, then enzyme part, other part gets activated. Okay, so it will produce the second messenger. So this second messenger, so hormone, its main action is to produce second messenger. Now the rest of the functions will be doing by the second messenger. It will initiate several reactions within the cells. So first, this response has to be made. This is called transduction. Transferring one kind of information to another kind. So it was hormone. But what is produced inside? Something else. Hormone is not entering. So in that way, signal is changed through the formation of second messenger. Hormone is the first messenger. It brought message from one organ, that is, say if it is insulin, from islets of Langerhans, so from beta cells, and it is, somebody told beta cells, you must release insulin. Who told? Because my glucose level in the blood is reduced. I am hypoglycemic. So if it is too much low, I may undergo hypoglycemic shock. So to avoid this, without glucose we cannot survive. So cells need glucose. So it will be, beta cells immediately will be stimulated and it will form insulin and it will liberate this hormone into the blood. Now through blood, the cell will come to all the cells, and it will help transfer of glucose from blood into the cell. Because without the help of the insulin, glucose cannot enter or cannot pass through the cell membrane. So cell membrane is having some channels. So through these channels are also protein. These channels, whatever the channel is, they are all proteins made up of several polypeptide chains. Suppose this is the channel. This is one polypeptide, this is another peptide, this is another peptide. So there is a binding site for the protein, for the hormone. So this hormone will come and bind there. Immediately these chains which are passing across the cell membrane, so there will be some conformational change. The peptide chains will produce some changes. As a result, the channel within this will be opened. Now there is higher concentration of, say, amino acid here, lower concentration is inside the cell. So there will be diffusion through it. Similarly, potassium ion, sodium ion, they have got channels. Without the channels, they can go in. See, one of the thing, very nice thing, potassium can travel easily inside they can pass through easily across the membrane, obviously through these channels. And what sodium ion also has to pass through these channels. But it can pass through relatively easier way in respect to sodium ion. But the size of the sodium ion is lesser than the potassium ion. Then where is the problem? The size of the potassium ion is bigger in respect to sodium ion. So what is the problem? Problem is the sodium ion is having a coat of water. And it doesn't want to liberate this coat. So as a result, its diameter becomes higher than the potassium ion. So it becomes difficult for the sodium ion to pass through so easily. Understood? That will be explained later. So transportation. Cell to cell communication. Is it needed? Is it needed? Suppose there are some epithelial cells or endothelial cells. Is it needed the information having in this should be transferred to the nearby cells?
Yes, yes.
It is needed. So its information should be communicated, and this communication is done by cell membrane. It has got some proteins. You will be knowing gap junctions, tight junctions, desmosome, cell adherence. So there are so many things you will be studying. So many things that will help. Some junctions. See the epithelial cells. We are swallowing something. Every day we are swallowing food. So these epithelial cells at the inner lining, they are always under stress. This skin is always under stress. So here the cells, epithelial cells, where the epithelial cells are open to the surface, like respiratory tract, like urinary tubule. Okay, such epithelial cells need to be adhered to each other tightly. Otherwise, when there will be stress, they will be splitted. There will be splitting. See, vitamin C. If somebody is suffering from vitamin C deficiency, there is bleeding. Why? Because they are the factor that controls the cell-to-cell binding. So when there is any deficiency, this binding will lose. So there will be bleeding. So that's why vitamin C is important to maintaining the epithelium. Okay. There are different kinds of communication, cell to cell communication. This is important. See, number one point. Number one, competency, the second part. What is written there? It's not intracellular communication and their applications in clinical care and research. This is newly added last year only. In 2020, it was not there. So we are expecting some questions from here. Since it is newly added, if you go through the CBME curriculum of previous year, you will not find this. You are having the syllabus, no? So why you are? Same, you are having the syllabus with you. I mentioned you have to study the cell-to-cell communication, intercellular communication. This is important for you. You must learn very nicely. Intercellular communication, cell-to-cell communication, it is done chemically, neurally, and some physical structure. One example of this is gap junction, physical structure, from this cell to this cell. So there will be cytoplasmic connections. So there can be exchange from this cell to the adjacent cells. Okay, so nothing is produced separately for this, so which will facilitate this transfer. Only thing, there is a channel, and here also hexomers, 6 polymers are there, and when they are closed, And this axon as part of this channel, this gap junction, part of this gap junction, the material donated by this cell and part is donated by this cell. Suppose this cell is having this and this cell is having this. So there is a gap in between. No, this is called extracellular fluid. But when these two membrane will come closer, this space will be reduced about 2 to 4 micron. So now this thing of the cell membrane, this is a special protein, this is also special protein of this, so they will be coming closer and join together. So this part belongs to this part and this part belongs to this part. So when they become activated, the pore is opened. Inside there is a channel and through this channel the information from this cell can go to that, from this channel to C. Example, heart. There are cardiac muscles. If one cardiac muscle contracts, the other muscle will also contract. This is called all or none law. If one contracts, the other will also. One cell is getting information for contraction. So how the other cells are getting? How the other cells are getting stimulation or contracting? Because information from this is going to the next cardiac muscle through some junctions. We call it intercalated disc. So immediately, what the information coming from SA node, electrical impulse is coming from SA node and stimulating the cardiac muscle cell. So this information, electrical signal will be passing to all the cells. As a result, all the ventricles will contract together. Otherwise, one muscle will contract, another will be relaxed, another will be contracting. So there will be haphazard. Okay, so because of this sort of connections, cell to cell connections, the whole heart, particularly the ventricle, contracts simultaneously. So there is such importance. So information should be transmitted from one cell to other. This is one way. Another way is beta cell of islets of Langerhans. It is producing something to act on distal cells, or say pituitary gland. It is anterior pituitary gland. It is producing TSH, thyroid stimulating hormone. So produced by pituitary cells of the pituitary. So this information should go to thyroid gland. How this information, this cell to this cell will go? Through endocrine system. So this endocrine system produces hormone. And this hormone has a target cell. Because those target cells are having receptors for this. These cells are not having any target for TSH. But they are having, these cells are having receptors for thyroxine. The hormones produced by the thyroid gland, when they will be secreted into the blood, their target organ is other cells of the body. So they are definitely there should be presence of receptors. So when TSH binds with these receptors, immediately its metabolic activity changes. So this way, see, through endocrine system, from one cell to another system, another cell is transmitted. Another way is, we call it paracrine system. He is a cell, he will regulate himself. He finds something abnormal within his cell, then he will produce some product and it will liberate into the surroundings, means extracellular fluid. Immediately, this product he is having on his surface cell membrane the receptor for this. So after liberating from this cell, it will again bind with the receptors, and now it will produce a different kind of changes within himself. So as a result, what will happen? The cell will be able to auto-regulate itself. Okay, understood? So this is we call auto-regulation. The cell will control itself. Suppose we are doing it. Introspection. There is a word, introspection. What is the meaning of introspection? Judge yourself. Whether you are doing some wrong or right, you judge. When you will be going to bed, you think yourself. So whatever I have done today, whether everything was right or wrong. So if you find, I have done something wrong, I had some altercation with my friend, but it was not proper. Actually, fault lies with me. This is called introspection. So by introspection, you can rectify yourself, isn't it? So if you introspect yourself, if you think I am studying well, okay. If you think I am not studying well, if you introspect that way, then we will start studying. So this is must, even in our life, introspection. So here also this cell undergoes introspection to correct itself. This is the one kind of communication, self-communication. Another kind of communication is, this is one cell, this is another cell. In between there is fluid, extracellular fluid. So he wants to, but there is no direct connection. So he wants to send some information to him, to her, to me. How he will send this? It is sent by paracrine. Autocrine, paracrine. So here, this information from here, no need of passing through the blood. Only it has to pass through the extracellular fluid. But for endocrine, I am the beta cell, my target cell is sitting there. Okay? So I will liberate the hormone, this will be carried by the blood and it will go to them. So for endocrine, this is a must. So this way we can make communication through autocrine, through endocrine system, through paracrine system. Another way of communicating from one cell to another is through nervous system. Example? Lot of examples you are having. You have studied neuromuscular junction. Another example, transmission through neuromuscular junction. Another is synaptic junction. Synapse means one neuron. It is transferring its information to the next neuron. So this neuron is having axon. So at the end of the axon, there is a sac-like structure. We call it synaptic knob. So here some chemicals are there. Say adrenaline or epinephrine. So when through the axon, nerve impulse will be coming and will entering into the synaptic knob. Immediately what happens? There will be release of epinephrine, and this epinephrine will be released into the gap, synaptic cleft. And here is the dendrite or cell body of another neuron. So this chemical, as say adrenaline, will cause depolarization. Yesterday I talked about depolarization. These cells are under resting condition. Okay, so its outside is negatively charged, also positively charged, inside is negatively charged. So this adrenaline will cause depolarization, means sodium, it will open the sodium channel. As well as sodium ion will be entering, and cell here this membrane will be depolarized. So when depolarization will be coming to a particular level, then action potential will be generated. Now this action potential will be traveling through dendrite into the cell body, from cell body to the axon, again to its terminal. So in this way, if I pinch here, it will be sensed by from here, my cerebral cortex. But this pathway, it has to cross so many neurons through these junctions. So information from here came here, from this cell to this cell. So this is the signal transfer from here to there. Okay, then we became able to understand how it escaped, what is happening at the periphery. So this is one kind of. Another thing, suppose my heart is beating very fast. Okay, very fast. So I am feeling uneasy. I am feeling there is tachycardia. What has to be done? The heartbeat has to be reduced. So immediately, hypothalamus could sense through its receptors. There are some receptors. They could sense. There is medulla, not hypothalamus, medulla and pons. There is cardiac regulatory centers lie there in pons, and so they will sense it has become increased. So the parasympathetic activity will be increased. Parasympathetic activity will be increased. So when this parasympathetic nerve supplying the heart will liberate acetylcholine from its nerve terminus. When this acetylcholine will make change in the cardiac muscle in SA node. So SA node is producing say 150 impulses in 1 minute. Now because of action of the acetylcholine, this will be reduced. So its activity of the SA node will be reduced. So as a result, less impulse formation, less number of impulse formation per minute. So according to the number of impulse produced in the SA node, the heart will be beating in that rate. So under the influence of acetylcholine, suppose SA node is producing 80 beats. So 80 impulses, 80 action potential per minute. So my heart also beat at that rate. So communication from the center to the heart came through this. So what is there? Electrical impulse. So it has produced some chemical, and this chemical actually produced changes in the target organ. So in this way, cell to cell communication is made. So what are the processes we learned? What are the cell to cell communication methods? What you learned from this? One is through gap junctions, another through some hormones, local hormones or paracrine hormones or autocrine hormones or endocrine systems. Okay? So they have, when they are liberated in their organ, so they will be liberated into the blood. So through this blood, these hormones will look for its target organs, receptors. So there it will bind and produce effect on the target organ. This is one way of communication. Paracrine, from the one cell to neighboring cells. No blood connection, only through ECF. And autocrine, self-regulation. So he want to regulate himself. Something abnormal thing produced. So this abnormal thing will come out. So it will again bind to the receptor, and this binding of this compound will initiate such reaction which will reduce the production of those compounds. So this is called autoregulation. You understood this? This is important for your, maybe this question I will cite during your January exam. January there will be exam on 100 marks. January. There will be 4 topics. One is already taught, hematology, and this is second. What you are to learn by heart. What are these? Cell junctions or intercellular communications. Functions of lysosomes. What are the difference between lysosome and peroxisome? There is difference. There should be some difference. So what are the difference? The structure will be almost same. They are look-alike, but they are functionally different. You are producing peroxisomes. You are producing certain things because of overactivity, and that will destroy your cell. Have you ever heard about antioxidants? Antioxidants? We eat antioxidants. No? So why we eat? Because of overactivity, some harmful products are formed in the cell, like hydrogen peroxide, like O negative, OS negative, OS negative. These are toxic. These are toxic oxidants. So we have to neutralize these oxidants to avoid the toxicity. So that's why we are taking anti. Can you name one antioxidant? No. So you are eating antioxidants.
Vitamin C.
This boy is very good. I must appreciate. He studies a lot. Very good. So I am expecting, there are 3 colleges in the city under this university. So you do something better in the exam, university exam. So this is the encouragement, motivation I am giving. So it is now your duty to be motivated and do hard work.